Hello everyone and welcome to our module on growth and genetics. What's shown on the screen here is a fairly classic microbiology graph of the number of bacteria present in a culture as a function of time. So if you were to perform an experiment in a microlab where you inoculated a flask of culture medium with a certain number of bacteria right about here, and then you watched over time how many bacteria were present in the flask, this is what you would see. There'd initially be a period where no bacterial growth would be occurring, meaning you aren't increasing the number of bacteria. Then after that lag period, you would see an exponential increase in the number of bacteria. Then it would eventually flatten out, and then finally you'd start to see a loss of the number of bacteria as bacterial cells die. So let's talk about what's happening at each phase of this bacterial growth curve. The first phase is called the lag phase, and so what's happened here is that you've inoculated the culture with a certain number of bacteria noted here on the y-axis, but no growth in the number of bacteria is occurring. Now, the bacteria may be growing in size, they're increasing the number of proteins, they're taking up some of the nutrients, but they haven't yet started to divide. Thus, as time goes by, you see a flat part of the curve because you aren't increasing the number of bacteria that are present in the culture medium. After the lag phase, you enter a phase called the growth phase. It's also sometimes called the exponential phase or the log phase. And what's happening here is now the bacteria have started to grow. So one bacteria is becoming two are becoming four, and thus you're getting an exponential growth in the number of bacteria that are present in your culture. And we can actually define an important term from the growth phase called the generation time or the doubling time, and this is the time it takes for the number of cells in the culture to double. And it's different for different bacteria. Some bacteria double more quickly, some take longer to double. And one other thing you should know about the growth phase is if you imagine the human body as your culture media and you imagine an infection as an inoculation that takes place at time zero, it's during this growth phase that antibiotics have their greatest effect. This is when the bacteria are multiplying rapidly, they're synthesizing lots and lots of new proteins and lots of new cell walls, and if you administer antibiotics, they will have the biggest impact right here on the growth phase. Sometimes you're asked on exams or in microbiology classes to do some calculations of how many bacteria you would expect to be present after a certain period of time. And you can do this with just some simple mathematics. So for example, if you inoculated the culture with 2 times 10 to the 5th bacterial cells, at the end of the lag phase, let's say that was 1 hour, you would still have 10 times 2 to the 5th bacterial cells because there's no increase in the number of cells during the lag phase. Then, after each doubling time into the exponential growth phase, you would double the number of bacteria. So after one doubling time, let's say that was 1 hour, so after another hour, you would have 4 times 10 to the 5th bacteria. And then after a second 1 hour period, another doubling time, you would then have 8 times 10 to the 5th bacteria. And this is the way you can do some simple calculations of how many bacterial cells will be present if you know the length of time for the lag phase and the doubling time for a given bacteria. The next phase is called the stationary phase, and what's happening here is that bacteria are continuing to grow, but they are also starting to die because there aren't enough nutrients left for them to grow as much as they grew in the log phase here. So you've got amount of death equal to amount of growth, and thus you get a flat portion of the curve where the total number of bacteria do not increase. And then finally you enter the death phase, and this is where the number of bacteria that are dying exceed the number that are growing, and therefore you start to have a fall in the number of bacteria that are present. So for the remainder of this module, what I'm going to do is talk about the way genes are transferred between bacterial. And you should know that bacteria often transfer genetic material, and this is very important for evolution, especially of things like antibiotic resistance. So as you know, sometimes an antibiotic will work for an infection for a period of time, but then it will stop working. And the reason it stops working is because the bacteria have evolved and now have genetic resistance to a given antibiotic. And there are 3 key mechanisms by which bacteria transfer genetic material, and they're called transformation, conjugation, and transduction. So the easiest method of gene transfer to understand is called transformation. This is when there is direct DNA uptake by a bacteria from the surrounding environment. So you can imagine if you've got a bacterial cell swimming around and there happens to be some DNA in the environment, maybe from another cell that died, or maybe it's in a lab experiment and we've purposely introduced DNA. Sometimes bacteria will take this up and incorporate it into their genome, and if the DNA they take up happens to impart an evolutionary advantage like antibiotic resistance, then this bacteria will now have a survival advantage. So taking up naked DNA from the environment allows for evolution of the DNA over time, It's a very useful technique in microlabs because you can introduce genes to bacteria for replication by just inserting them into the culture media and allowing the bacteria to take them up over time. The next method by which bacteria transfer genetic information is called conjugation, and this is where one bacteria cell directly transfers genetic information to another cell using an appendage called a pilus. Now you may know that some bacteria like E. coli have pilus that they use to attach to surfaces. These are different kind of pili called sex pili, and two bacteria can attach via sex pili and exchange genetic information. So there has to be physical contact of the two organisms. And another thing that you have to know is that the DNA here are transferred via plasmids. So let's talk about what plasmids are. So plasmids are small DNA molecules within a cell, and they're physically separated from the chromosomal DNA. So if you look at this picture down here, the red circle is the bacterial DNA, but these little blue circles are plasmids. They're little bits of genetic information floating freely inside the cell, separate from the DNA. And this is how bacteria are going to transfer genetic information through conjugation. The plasmids can replicate independently. A lot of times they contain genes for antibiotic resistance or for toxins, and they can be transferred from one bacteria to another. This slide is a depiction of how conjugation takes place. So we have a bacteria on the left side of the screen that has a plasmid, and we have one on the right side of the screen that does not have the plasmid present. A sex pilus is extended from the donor to the recipient, and they connect, and then genetic material crosses from one to the other. And then the two bacteria separate, and now the plasmid is present in the original bacteria as well as in the new one. There's a classic microbiology experiment that's conducted to prove that conjugation takes place between two bacteria, and you should definitely know about this because it can come up on exams. So let's imagine that we have strain A of a bacteria, and it requires amino acid X for growth, but it doesn't need amino acid Y. Strain B is the opposite. It requires amino acid Y, but doesn't need X. So we put them together on a culture media without either X or Y, so neither of them should be able to grow because we've deprived them of the elements that they both need in order to grow. And then we add something called DNase to the medium. So DNase is an enzyme that breaks down DNA, and it's going to degrade any DNA that's found freely in the culture medium. After we do this, the bacteria still grow. So how do we explain this? Well, what this means is that the ability to grow without X or Y must have been transferred between bacteria. And it cannot have been transferred by transformation. So genetic material cannot have been dumped into the growth medium and then picked up by another bacteria by transformation because DNase was present. It would have destroyed any leaked DNA. So this proves that the way the genetic information must have been transferred from one bacteria to another is through conjugation. They must have attached and transfer the DNA without allowing it to be exposed to the DNase enzyme that was present in the culture media. Make sure you understand this experiment and why it proves that conjugation takes place instead of transformation. There are some bacteria which we call high-frequency strains of bacteria. And what these bacteria tend to do is take plasmids, like this blue circle right here, and incorporate it into their own DNA. So I've drawn here a blue plasmid that's now been incorporated into the bacterial DNA. The reason these strains are important is because when they conjugate with another bacteria that doesn't have the plasmid, they tend to transfer their entire genome along with the plasmid, or at least portions of it along with the plasmid. So the reason this matters is because we can use it to map genes. It takes time for conjugation to transfer the plasmid and the entire gene from a high-frequency strain to another one, and we can interrupt it at various points in time. And then we can see which genetic material has transferred. So what generally happens is the plasmid is the site of origin for genetic material. So the plasmid gets transferred and it drags along portions of the bacterial gene along with it. So we can interrupt that process. And for example, if we see the plasmid portion right here has been transferred and then we see a gene for antibiotic resistance, we know that that gene for antibiotic resistance must be very close to the plasma insertion site. We can then allow some more transfer to take place, and we may see further bits of the bacterial genome transfer, and then we can map what those are. And this is the way we can get an entire map of a bacterial genome, and that is the real utility of high-frequency strains in a microbiology laboratory. The third method of transfer of genetic material between bacteria that we'll talk about is called transduction. And this is transfer of DNA via a bacteriophage, which is a virus that infects bacteria. So the virus picks up DNA and then transfers it to another bacteria. There are two subtypes of transduction that you should know about, and the first type is called generalized, and that's the easiest type to understand. This is when the virus infects a bacteria, it multiplies inside the bacteria and randomly picks up host DNA, and then the host DNA gets transferred to other bacteria. And there's a depiction of that on the screen here. So we can see a virus with some purple genetic material inside it infecting a cell with green genetic material. Once the genetic material's inside the cell, the purple and green stuff all start to multiply together and new viruses are created. Some of them contain the purple DNA of the original virus, but some contain the green DNA of the bacteria. And then when enough viruses are present, they rupture the cell and they all spill out. One of the new viruses carrying green material from the original host bacteria that was infected by the virus can then infect another bacterial cell and stick its DNA inside, and it can potentially become incorporated. So this is generalized transduction. This virus is infecting the cell and randomly picking up pieces of the original cell's DNA and creating new viruses which go on to transfer that material to another cell. You can see that the net result here is that some of this green DNA of the original bacteria has been transferred to another bacteria via the bacteriophage as the vector. Specialized transduction is the second type of transduction, and this is the transfer of very specific genes from a bacteria to another. This happens when the virus DNA inserts into the host DNA. This is a process called lysogeny, which I'll talk about in a minute. So that viral DNA is going to live in the bacterial cell's genome for a while. Later, when the bacteriophage DNA is excised, it's packaged into a virus with specific host DNA, the DNA that sat right next to the viral DNA when it was incorporated. And then this host DNA will then get transferred to other cells via a viral infection vector. So I mentioned the process of lysogeny, so now let's talk about the difference between lytic bacteriophages and lysogenic bacteriophages. Transduction generally happens in two ways. The first way is called the lytic cycle, and that's that generalized transduction that I described 2 slides ago. Nuclear material enters the bacteria from a virus, it multiplies and lyses the cell, and it releases progeny viruses. This is called the lytic cycle because it lyses the bacteria that gets infected. The second method of transduction is called the lysogenic cycle. This is where nuclear material enters a cell from a virus but then incorporates into the host DNA. It may later become excised and then it will enter the lytic phase and it can kill that bacteria, but it also may just live inside the bacterial DNA for a long time. Phages, which remember are viruses that infect bacteria, phages that replicate only via the lytic cycle are called virulent phages. Phages that replicate using both the lytic and lysogenic cycle are called temperate phages. So this slide shows how lytic phages operate, and they operate very similar to the method we described a few slides ago for generalized gene transfer. The virus infects the bacteria, uses cellular machinery to reproduce, and then lyses the cell, and you usually get a generalized transfer of genes. So this is that same depiction from a few slides ago. The purple viral material is going to be delivered to a bacteria that contains green genetic material, and then we're ultimately going to get rupture of the bacterial cell with new viruses, some of which contain the original bacteriophage genetic material, and some which contain the bacteria's DNA or RNA itself. A lysogenic phage operates like I've described on this slide here. So the virus infects the bacteria, but it incorporates the phage DNA into the bacterial DNA, and it can actually remain dormant for long periods of time. Sometimes a trigger like UV light will induce genome excision, and this is sometimes used in microlabs to get the genome to pop back out. And when it gets excised, then it can result in a lytic cycle and the release of phage particles like we described on the last slide. But notice here that I've drawn a picture of a virus sticking some blue genetic material into a bacteria, and then that gets inserted into the bacterial genome. This is called a prophage region of the genome, and this can stay here for a long time, and this is a mechanism by which a new bacteria can acquire abilities that it didn't previously have. The reason lysogeny matters in medicine is because the genes for some bacterial toxins are transferred to non-toxic strains via lysogeny, and one of the best examples is diphtheria. Many strains of Corynebacterium diphtheria are non-toxic, and in fact the gene for the diphtheria toxin is not part of the bacteria's normal genome. In order for a Corynebacterium diphtheria cell to become toxic, it has to get infected by a phage called a corynophage, and this will impart the ability to synthesize the toxin, and then the diphtheria bacteria cell will become toxic. And I've listed on this slide here some of the toxins that are imparted by viruses to bacteria via lysogeny. So diphtheria toxin is one of the best examples. Also erythrogenic toxin of strep pyogenes, which causes scarlet fever. the Shiga-like toxin of E. coli, also cholera toxin, and botulinum toxin. The last topic I'll mention in this module is that of transposition. So transposons are DNA segments within bacterial DNA that can be excised and then reintegrated into new locations. They're sometimes called jumping genes. The thing to know here is that once they're excised, they can also sometimes be moved to a plasmid, and this is a mechanism of transfer of resistance to antibiotics for many bacteria. So the way this works is bacteria number 1 is resistant. A transposon segment carries the resistance gene, so this segment is capable of jumping around the bacterial genome. At some point in time, that transposon may jump and just stay removed as a plasmid, and then the plasmid can get transferred to other bacteria and transfer the ability to resist antibiotics. And that concludes our module on growth and genetics.